Okay, let's get started. Good morning, everybody. In the last lecture we had together, we talked about thermodynamics, and the whole theme or the major theme of the lecture was how do we find out, how can we measure, how can we figure it out, which processes will spontaneously happen and which will not. We talked about entropy, all the different laws, etc. Can you hear me? Can you hear me at the back? Yep. Okay. But I think I mentioned in the lecture that thermodynamics is just a part of the picture. It tells us which processes are potentially spontaneous, that want to happen. But it doesn't necessarily, well, it doesn't really say anything about whether they will happen in actuality, whether they will actually happen. Why is that? What is the problem? Okay. Yeah.
Kinetics? Kinetics?
Okay, so even though a process, a reaction or whatever may be thermodynamically favorable, okay, so it wants to happen, it should be spontaneous, some of those processes, actually a lot of them, never actually happen because they are too slow or the activation energy is too high or whatever you want to call it, okay? So kinetics is the different part of the story, the other part of the story, and we need both of them in order to be able to describe what's actually happening in our cells or in our test tubes or what have you, okay? You had a lecture on catalysis and enzymes, which talks about kinetics. So what are catalysts? What do they do and what do they not do? Okay, I'll probably have someone else suggest. Okay, what are catalysts? What do they do in general?
Decrease the activation energy.
Okay, they decrease activation energy and
without self being used.
Okay, they're not consumed in the reaction. Correct.
Speed up the reaction.
Okay, they speed up the reaction. What is it that they actually speed up? You're right, huh? The rate of reaction, correct. Okay. So what is it they do not change?
The amount of substrate, or
The amount of
substrate that's being,
Amount of product that's being produced.
the equilibrium.
Okay, they do not move, they do not change the equilibrium of the reaction. Okay? So where the reaction, yeah, do you want to?
Do the enzymes increase it by binding to the other catalyst? Like binding to the particles which are moved, so therefore they increase the velocity, so they collide quicker which is still
okay.
equilibrium, it's just they move faster.
Correct. Yeah, so you start talking about the molecular mechanism behind it. It's usually not speeding up the collisions, it's allowing them, as you correctly said. But the important thing is the equilibrium, so the result of the reaction, the thermodynamic result of the reaction is going to be the same regardless of whether there is a catalyst or an enzyme. Okay, this is a very important thing. So kinetics and thermodynamics are two distinct things. Okay, one talks about equilibria, talks about how the reaction is going to move, which way it's going to move, where it's going to stop. And kinetics talks about how fast it's going to happen. So catalysis, as you correctly said, or catalysts, just speed up the attainment, the achievement of the equilibrium. That's all they do. Okay? So if there is a reaction which is thermodynamically unfavorable, it's not going to happen no matter how much catalyst you add. Okay? Very important distinction. Now, in today's lecture, we're going to follow up on the enzymes lecture. We're going to talk about inhibitors, inhibition of enzyme catalysis. Why is it important? Well, because in pharmacology and in medicine in general, you're going to be using, as doctors, you're going to be using a lot of enzyme inhibitors as drugs. Okay? Because enzymes are essential for a lot of processes, for many processes in our cells, in our body. So it's advantageous in certain situations to inhibit a function of an enzyme in order to produce an effect that we want to see in a patient. Okay? So today's lecture is going to be about inhibition of enzymes, how we can inhibit them, what types of inhibitors there are. And in the second half of the lecture, in the second part of the lecture, I'm going to be talking about a few examples of enzyme inhibitors that are actually used in medicine and in some other areas. And I'm going to talk a little bit about how they actually work and what they do. Okay? So it's going to be about inhibition. In order to be able to talk about inhibitors, I think it's probably a good idea to review a little bit enzyme kinetics as a whole. So you in the enzymes lecture, you talked about one model of enzyme catalysis, of enzyme kinetics, which is
Materials constant.
okay, Michaelis and Menten model. Okay, why do I call it a model? It's really just an abstraction of one type of enzymes. Okay, it's the simplest one, and that's the reason why you taught why we told about it. But it does not describe, and that's just a kind of disclaimer. It does not describe all the enzymes because there are many variations. But Michaelis-Menten kinetics is the simplest model, and that's why we're going to build on that. Okay, it's actually very useful for the description of inhibition as well. But I just want to say it does not really describe all the enzymes because some of them actually work in a different manner. Right, so Michaelis-Menten model. Before going in the in the mathematics and some of the graphs, what is the model based on? What is the rationale? How do we imagine that enzyme catalysis actually works? Yeah?
It basically tells you the affinity of the enzymes to the substrate.
Okay, it talks about the affinity. What are the abstracted reactions going on during enzyme catalysis?
Forms a complex with the substrate.
Very good. So we start with some enzyme molecules and some substrate molecules, which associate together, depending on the affinity, as we'll see in a minute, into an enzyme substrate complex. And very important, that's a reversible reaction, okay? Sometimes it's more reversible, sometimes it's less. But in theory, at least, it can go back. It's very important for the model, okay? So the enzyme substrate complex can also dissociate back into enzyme and substrate. What is the other possibility for the enzyme substrate complex to do? Yeah?
Irreversibly proceed to product and enzyme.
Okay. Okay, usually there's one step more, which is the enzyme product complex, but I'm going to leave it out just to make it simpler. Okay. Now, did you say irreversibly or reversibly?
Irreversibly, yeah.
Okay. Yeah. I mean, I don't want to go into too much detail. Sometimes it is assumed that this is practically irreversible to make the calculation simpler. But this is the actual description of what goes on. Okay. So this, in theory, is also reversible, so it can go back as well. Okay. And depends on the equilibria and the thermodynamic considerations for that. But we're not going to be talking about thermodynamics today. We're going to be talking about kinetics. Right. So this is kind of the abstraction. This is what we think is going on in the in our generic enzyme. Okay. Now, and we're going to be using this to describe inhibition as well. That's why I wrote it down. Right. We have a mathematical expression of the model, which is the Michaelis-Menten equation. Can somebody from the back tell me what the equation looks like? Yep. You don't have to raise your hand. You can just shout. Okay.
The velocity or the rate of velocity is equal to Vmax times the concentration of substrate over the Km plus substrate.
Okay. What is Km? Yep.
K negative 1 plus K 2 divided by K1.
What are K1 and K2?
The rate of transfer between the states in that form.
Okay. Okay. Okay, so those are the rate constants for that. I wasn't really asking, you're right, but I wasn't asking about the how it's derived. But what is it? First of all, what is it called? It's not just Km, it's got a... Okay, it's Michaelis constant, very good. And what is it? What is it?
Vmax divided by 2.
Is it equal to Vmax divided by 2? Okay, who thinks it's... raise your hand if you think it's Vmax divided by 2, that it's equal to that. Okay, some people think that. Okay. Is there anybody who has a different idea? Okay. It's, you don't have to raise your hand and wait for me to ask, okay? You can just shout. But if you just like wave at me or something so that I know who's talking, okay? But you don't have to wait for me to, yeah?
It's the substrate concentration which corresponds to the 1/2 of the maximum.
Okay, so the description, if not necessarily the mathematical definition of it, the description of Michaelis constant is it's the concentration of substrate at which the rate of reaction is going to be equal to V max divided by 2. Okay, so half maximum rate. Okay, is that familiar? Does everyone? Yeah, it's a very important definition or description, so you should really know that. Okay, Km is equal to the concentration of substrate at which the rate of the enzyme catalyzed reaction that we're looking at is going to be half maximal. It's going to be half of Vmax. Okay, I'm going to draw the graph in a second so we can look at it. Okay, that's a very important definition. You should really know it. Okay, so we can express this kinetics using a graph. What are going to be the axes in this graph? You've probably seen it before.
Velocity and concentration.
So which one's velocity? Which axis? Okay. Okay, so this is the rate of the reaction, and the x-axis is the concentration of the substrate. Okay. Can you all see it when I draw it down there? Yep. Well, especially for you. Yeah. Right. So what is the relationship? What does it look like for an enzyme that actually follows this kind of kinetics? What does the relationship between the two variables look like? If we increase substrate concentration, what happens to the rate? It goes up, which kind of makes sense. Does it go up in a linear fashion or a non-linear fashion?
Hyperbolic?
Okay, so the shape is hyperbolic, correct? Okay, so strictly speaking, you're right, but strictly speaking, it's non-linear. But it's true that to a certain extent we can assume that it's almost linear, okay, in the beginning. Okay, which is actually quite useful for some calculations because it makes them a lot easier. Okay, so the beginning of the curve we can sort of say, oh, it's almost linear. Okay, but it's not linear. It's actually not linear in any part of the curve. Okay, you can just say that it's not very far off. Okay, right. So in the equation we have basically two variables that are linked, which is rate of the reaction and the concentration of substrate, and we have two parameters. Okay, two constants basically. Okay, which describe the specific reaction. One of them is, as we said, is Km, and the other one is Vmax. Okay. What does Vmax depend on? What is it dependent on? Okay. So it depends on the amount or concentration of enzyme in our reaction. Okay, this is very important because the more enzyme we have, the higher Vmax is going to be. Okay? Fairly important thing. Again, it's going to be useful when we talk about inhibition. Okay? So it depends on the enzyme amount or concentration or number or what have you. Does it depend on anything else?
Substrates. Amount of, I guess, the amount of substrates that should collide with the enzymes, because it's a two-way thing. Like an enzyme a substrate bind to collide with the enzyme to give you something else. So it's both of them, the concentration of both of them, I guess.
Doesn't it also depend on Km because of the affinity? On the temperature?
It depends on temperature, absolutely. It does not depend on Km. The two parameters are actually independent, and we'll see in a minute how that works, okay? So they're not dependent on each other. Those are two independent parameters that describe a specific enzyme catalyzed reaction.
Can I ask why? Because if the affinity is higher, wouldn't that be faster then?
Well, at a given, okay, let's go one step back, okay? What is Vmax? What is Vmax? What is it?
The speed at which the enzyme is completely saturated.
Okay, that's true. But when does this situation occur? In which situation do we get a complete saturation of the enzyme?
The maximum rate of enzyme if we have enough substrate.
Okay, what is enough substrate?
more substrate than the enzyme can...
How much more?
Just more.
A little bit more. Okay.
Product of it's A times B, the product of
Okay. This is a mathematical abstraction. Okay. When do we reach Vmax?
Never.
Well, not really never, but
Equilibrium?
it has nothing to do with equilibrium. Okay? Kinetics, thermodynamics, completely different things. Well, not completely, but let's assume they're completely different things. Okay? One tells us about rates, and the other one tells us about when equilibrium is reached. Okay? Not related in any way.
When the derivative equals 0, of the mathematically speaking, where it equals 0.
It will eventually, yeah. But I'm asking about what do we need to get there?
A specific amount of substrate.
Yeah. What amount of substrate?
The amount of substrate that the enzyme can handle.
Higher than that.
Okay. Okay.
When all the active sites are...
Sure. But when does that happen? When all the active sites are occupied?
So we keep putting substrate...
Okay. How long do we need to keep putting more substrate? Okay, so what is the concentration of substrate at Vmax? It's infinite. Okay? At infinite concentration of substrate, that's when we get Vmax. Okay? So when somebody said it's never reached, well, in practice, yeah, it may be never, you know, again, this is a mathematical description, but in this description, we only reach Vmax at an infinite concentration of substrate. Okay? This is very important. Okay, now back to your question. You said, well, doesn't it depend on the affinity? Well, it doesn't, because at an infinite concentration of substrate, it doesn't really matter how you know, if the enzyme really wants to bind the substrate or not. There's just so much substrate, there's infinitely more substrate, that the Vmax is going to be the same.
But the enzyme just has to bind to that? Or?
Well, pretty much. I mean, we assume that if the substrate catalyzes the reaction, there is at least some affinity. It's non-zero. And if it's non-zero, then it doesn't matter what it is. At infinite concentration of substrate, it's always going to be maximal.
But that's only when it's pure? Like when there's no other substrate?
Oh, sure. Yeah. We're just assuming we only have enzyme and substrate. Of course, if there, we'll see when we talk about inhibition, that's where we see it's a bit more complicated in that respect. Okay? So Vmax and Km are not related. Okay? Two different parameters. Yep.
You said infinite concentration.
Yep.
But at infinite concentration, what will it collide with at that case? Like,
It's a mathematical abstraction. Okay? We use a super simple model. This is way simpler than what's actually happening. Okay? This is a huge simplification. So in this whole lecture, well, at least for most of it, don't think about individual enzyme molecules colliding with individual molecules, okay? We look back at the whole test tube, and we look at what's going on in these billions and trillions of enzyme molecules and substrate molecules, okay? This is what we're describing, okay? Describing it at the level of one enzyme, that's a completely different description. It's much more complicated, and we're basically not going to be talking about it at all. Okay, so this is a description of the bulk solution, of the bulk reaction. That's what we're looking at. Okay, so even though this idea that things are colliding and you know happening, it may be useful, but be aware that we're just describing a bulk stuff going on. Okay, it's a statistical summation of the different reactions that actually go on in the test tube. Yeah.
I don't know if this is relevant, but I'm thinking if it's infinite in the amount of substrate, then when does it reach the negative feedback?
No negative feedback here. Okay, our hypothetical enzyme that we're describing has absolutely no regulations, nothing. It's just a molecule that sits there and performs our reaction. That's all. Okay, so no feedback, nothing. Okay, no phosphorylation, nothing. Okay, so does it make sense why Km and Vmax are independent? Okay, it does not depend on the affinity of the enzyme what Vmax is going to be. It depends on the enzyme amount, it depends on temperature, and some other things as well. Okay, that's what it depends on, Vmax. All right, and as we said, Km is, if we take Vmax divided by 2, that's the concentration of substrate at this rate. So this was a very quick review of enzyme kinetics. And we're now going to be using it to describe the different types of inhibitors. And I think that the different classes of inhibitors or different types of inhibition were already mentioned in the enzymes lecture. Can anybody remember what they are? Very good. So we have competitive, non-competitive, and uncompetitive. Sometimes you can see a-competitive or anti-competitive. It's all the same. Uncompetitive, a-competitive, anti-competitive. Okay? Those are going to be the three types of inhibition we're going to be talking about. This classification of types of inhibition is based on the relationship between the binding of the inhibitor and the binding of the substrate. Okay? And that's what we're going to spend most of the rest of the lecture talking about. But there is another way to describe or to classify, to categorize inhibitors, and that's based on the duration of inhibition. Does anyone know that? How?
Reversible and irreversible.
Very good. It's reversible and irreversible. We're not going to be talking about it basically at all until the very last part of the lecture, but I just want to say that this is a continuum. Okay? There's no way to basically distinguish between a reversible and an irreversible inhibitor. Okay? All the inhibitors bind to the enzyme and stay there for some amount of time. Some of them for a longer time, some of them for a shorter time. Okay? So this is a continuum from completely reversible inhibitors that bind very, very quickly and then are dissociated, all the way to inhibitors that once they are stuck to the enzyme, they are stuck forever, pretty much. Okay? But it's a continuum, so we can't really say, well, this is reversible and this is irreversible. Well, we can say that, but it's usually only said for practical reasons. Okay? So if we know that the lifetime of an enzyme molecule is 1 day, then if the inhibitor stays there for longer than 1 day, well, then it's irreversible because the enzyme is going to be destroyed before the enzyme inhibition is released. Okay? But this is just it's arbitrary if we say this is irreversible and this is reversible. Yeah.
And how is it related to bonding? Because it said somewhere that it's covalently bonded, non-covalently.
Yep. We'll talk about it a little bit when I talk about specific examples, because then we'll see the different kinds of bonds, what happens. Okay? Because even covalent bonding can be reversible. Okay? So this is just, it's a continuum, reversible to reversible, yeah?
Are these reversible or unreversible?
We will assume in all of these that it's completely reversible inhibition. Completely reversible. Okay? For irreversible inhibition, the description is much more complicated. Okay? But I might actually mention it in some of them that, yeah, anyway. Right, let's have a look at these three categories. As I said, these types are based on, or the distinction between these types is based on the relationship between the binding of the inhibitor and the binding of the substrate. Okay? That's all we're interested in now. So how does the binding of substrate or the binding of inhibitor influence the binding of the other one? Okay? How does the binding of the substrate influence the binding of the inhibitor? How does the binding of the inhibitor influence the binding of the substrate? That's all we're interested in this analysis. Okay? Yeah, does it make sense? Raise your hand if it makes sense. Very good. Okay, let's start with the first one, competitive. Well, it's already in the name. So what happens in competitive inhibition? Okay, let's not talk about what happens to Vmax. Just let's describe it.
They both compete for the same enzyme, the inhibitor and the substrate. They both, whichever collides first with the enzyme, that's the competition.
Okay, so in competitive inhibition, the inhibitor and the substrate compete for binding to the enzyme. Okay? Most often, they bind to the same binding site. Not always, but most often they bind to the same binding site. It's usually the active site, but it doesn't have to be. Okay? Which explains if they bind to the same binding site, well, either substrate is bound or the inhibitor is bound. They can't be bound at the same time. Okay? That's why they compete for the binding. Okay? So in this model of enzyme action, or whatever, enzyme catalysis, if we start if we add a competitive inhibitor to this reaction to start with, okay, what's going to happen? Is there going to be any side reaction?
Yeah, IS as well as ES.
Hmm?
Yeah, IS as well as ES.
Really, is it going to be IS?
It's going to be IE or ES.
Yeah, IE, I think.
Very good. So we're gonna get a side reaction which produces a complex of the enzyme with an inhibitor, which is catalytically inactive. Okay? Again, notice that it's a reversible. In this model, we assume that it's a perfectly reversible inhibition, perfectly reversible binding. Okay? So in addition to our reaction going as it was before, there's a side reaction which forms an enzyme inhibitor complex, which is catalytically inactive. Okay. So what will happen to the kinetics? When you think about it, spend a little bit of time thinking about what will happen, okay? I know some of you know the answer already, okay? But I think it's quite useful to think about what might happen in this case. What will happen to the two parameters? So what will happen to Vmax? Sorry. Not the ones that know the answer, okay? The ones that thought about it. Because I will then ask you to explain why that is. Sorry, who's speaking? Yep.
It is not going to be reached, Vmax.
Okay, so Vmax for the inhibited enzyme is going to be lower than Vmax for the uninhibited. Is that what you're saying?
Yes.
Okay. Okay. Raise your hand if you agree with that Vmax is going to go down. Okay. Okay. About, yeah, okay, maybe a fifth of people. I'm sorry. Okay, who thinks it's going to be increased? Nobody? Okay, nobody thinks it's going to be increased. Who thinks it's going to stay the same? Okay, about the same amount of people. Okay. Okay, so out of those who think it's going to stay the same, explain to me why it's going to stay the same. Okay? Well, it can't. Sorry, did you not hear that? Okay, so the explanation was that it's going to stay the same, but it's going to shift to the right. Okay, we're talking about Vmax, so I'm not entirely sure how it can be shifted to the right, because it's just a number. It's just a number. Okay. Okay, let's stay with Vmax, okay? Then we're going to talk about the other parameter that describes it. So what happens to so you said that Vmax is going to stay the same, right? Okay, can somebody explain why it's going to stay the same? Yeah?
It's basically because the same, like the enzyme is going to bind to the same number of either substrate or inhibitor in this case, so it's going to stay the same. Like,
the same amount of substrate.
If we have substrate or inhibitor, like if we have 5 enzymes, the 5 of them are going to bind, but it's going to differ whether it's a substrate or an inhibitor in this case. But the same number of enzymes are going to bind to the same thing.
Why? There's the inhibitor there.
Yeah, but it's going to bind at the end of the day. But we only care about binding. We don't care about whether it's going to.
Well, why is it going to bind? There's still the inhibitor there.
It's a chance.
Why is? Yeah.
I'm thinking that it will bind eventually. It will take time, but it will bind because they're competing at the same site.
Yeah, but if you have many enzymes, some of them will bind the substrate, some of them will bind the inhibitor.
Yeah, but the ones they are binding to the substrate will.
Yeah, they will definitely work.
Yeah.
But what about the ones that bind the inhibitor? They're not going to work, right?
It depends on the concentration of substrate is infinitive, isn't it? So if you just add more and more and more, then in the end there's going to be one inhibitor molecule per 1,000. So.
Very good. Okay? This is why in the beginning I kept stressing that Vmax is reached at infinite concentration of substrate. So imagine that we added 1 micromolar inhibitor or 1 millimolar, it doesn't really matter. We added a fixed amount of an inhibitor. Okay? And then we keep adding more and more and more and more substrate. At infinity, there's going to be infinity times more substrate than the inhibitor. Okay? So the enzyme is not going to even see the inhibitor because there's infinite times more substrate.
Yeah, but it's still still a chance.
Yeah, but the chance is zero to infinity, basically, or 1 to infinity. Because there's infinite times more substrate.
But if you don't have infinite...
Yeah, but we do, because we're talking about Vmax. Okay, raise your hand if this explanation makes sense. Okay, this is a very important concept. So if the substrate and the inhibitor compete for the same binding site, at a fixed concentration of an inhibitor, the Vmax is going to be the same because at an infinite concentration of substrate, there's always going to be infinite times more substrate than the inhibitor. I can still see that some people are a bit puzzled by it. Raise your hand if you want me to explain it once again and more slowly. Okay, quite a lot of people. Good. So, once again, Vmax is only reached when there is an infinite amount of substrate. Okay? So, imagine that we start with 10 molecules of inhibitor, 10 molecules of a competitive inhibitor. Sorry, I'm showing 5, but I mean 10. Okay? We start with 10 molecules of competitive inhibitor. And in the beginning of our analysis, we have zero concentration of substrate. There's no substrate. What is the rate of the reaction going to be?
0.
Okay? It's going to be 0, obviously. Then we keep increasing the amount of substrate. Okay? We add 5 molecules of substrate. Okay? What is the rate going to be? It's going to be higher, obviously, than 0, because we have some substrate, and the substrate and the inhibitor are going to be competing. We have 5 molecules of substrate, we have 10 molecules of inhibitor. Okay? The rate is going to be probably still very low, but there's going to be some, because they're competing. Okay? Then we add 10 molecules of substrate, and we still have 10 molecules of an inhibitor. Okay? Again, they're going to be competing. The rate is going to go up again. Yeah? But there's going to be a competition again. Okay? Now we keep increasing the amount of substrate, and we only have 10 molecules of inhibitor all the time. And we keep increasing. So we have 1,000 molecules of substrate, we have 1,000,000 molecules of substrate, we have 1,000,000,000,000 molecules of substrate, we have infinite amount of substrate. Okay? Now, assuming, it doesn't have to be true, but I'm just trying to make it simpler, assuming that the likelihood of binding that the binding of the inhibitor and the substrate are the same, if we have an infinite number of molecules of substrate and only 10 molecules of inhibitor, the enzyme is only going to bind the substrate, okay? Because it's infinity to 10. Okay, as you said, it's based on chance, and in this case, the chance is going to be 100% binding substrate. Okay? So Vmax is going to be the same. Okay? Good. What about Km? What's going to happen to Km? Okay, let's try someone else. Okay, yeah. Okay, why is it going to increase? Okay, so, well, the enzyme has affinity to the substrate, but that's fine. Why does it have a different affinity now? Okay, okay. Yeah, that's based on the description, and you're right. Yeah, absolutely. I'm probably going to use the description that was before. I think it's easier to understand. So the Km describes the affinity, let's say the probability of binding the substrate. Okay? The higher the affinity, the higher the probability. Does it make sense? If we imagine that Km describes the probability of binding the substrate, or rather than binding the substrate, of forming the complex ES. Okay? Let's assume that Km, well, it's sort of true, that Km describes the probability at a given situation that ES is going to be formed from E and S. Okay? Can we agree on that? Not really, but yeah.
If you're increasing Km, so if we're having competitive inhibition,
Yep.
so the curve shifts.
Yep.
When you're increasing Km, then it takes you more concentration to reach the same
Yep.
velocity, correct? So how does that increase the probability of the affinity? How does that do with the probability? So how does it increase the affinity? Because the affinity should stay the same, but...
No, the affinity, the apparent affinity actually decreases.
Right. Okay.
Yeah? Because Km is inversely related.
Sorry.
Okay? So increasing, that's probably an important point as well. Increasing Km means decreasing affinity. Okay, I think you covered that in the previous lecture, but that's important. Okay, so there was a suggestion that Km increases in this reaction, and the reason is that the affinity of the enzyme for the substrate is going down, that there's less affinity. I can see a lot of you are now really confused.
Can you repeat that once more? Sorry.
Okay, so there was a suggestion from the back. I'm not saying that it's true, but it is, that in the competitive type of inhibition, Km is going to go up, which means that the affinity of the enzyme is going to go down. Okay, because they're inversely correlated. They work inversely. If Km goes up, I'll explain in a minute why. Well, I wasn't planning on, but I can explain why that is so. But Km is basically if it's going up, affinity is going down. There's less affinity of the enzyme to bind it. Okay, so. And the explanation for that is that if we assume that the affinity is the probability of forming ES, then in the presence of an inhibitor, that probability is going down because we have another reaction that is sucking away the enzyme. Okay? So the probability of forming ES is less, which means less affinity for the substrate, which means higher Km. Okay? So in competitive inhibition, we get the same Vmax, and we get higher Km. And the curve is going to look something like this. Okay, it's going to be shallower. I mean, it's not quite hyperbolic, but anyway. Okay, the curve is going to go up more slowly, but in the end, at infinity, it's still going to reach Vmax. Okay, at infinity. Yeah.
Why doesn't it have the same shape but just shifted to the right?
Well, you have to start at 0. Because at 0 substrate concentration, you're going to have 0. So you can't shift the whole curve. Oh, you're saying that basically that there should still be, at some concentration of substrate, there should still be 0 velocity, 0 rate of reaction.
Yeah.
Well, it won't really, because the two compete, okay? So it's not like the inhibitor just sits on those molecules. And also, we assume that we have more molecules of enzyme than we have of the inhibitor, okay? Which doesn't actually have to be. But anyway, it's not shifted, okay? The shape changes. Okay, we start at 0, 0 anyway. Okay, at least in this model. Okay, does it make sense? Yeah? Okay, very good. So if we take Vmax divided by 2, which is still the same Vmax divided by 2 because Vmax stayed the same. So we'll see that in this case, Km for competitive inhibition is going to be larger than Km for the native enzyme. Okay? Now, to make things more complicated, I'll go back from the bulk description to the individual enzyme. Obviously, the individual molecule will bind the substrate with the same affinity. Okay? The enzyme doesn't know, the molecule of the enzyme doesn't know that there's also inhibitor floating around. Okay? So if it encounters a substrate, it will bind it with the same affinity. But the trouble is, it can also encounter the inhibitor, and it will bind with some affinity. And those two effects combined mean that the apparent affinity of the enzyme, the bulk affinity of the enzyme, is going down.
So it's more like an average.
Yes. Yeah? As I said in the beginning, we're looking at bulk properties, okay? Not the individual enzyme molecules.
Can you say that again, please?
Which part?
30 seconds ago part.
Okay. I don't know what I said. That for the individual enzyme molecule, it doesn't really change in any way. Okay? The enzyme molecule is still the same. The only thing that changes is that the likelihood of binding a substrate is less because it can also bind the inhibitor. There's more to bind, which is not a substrate. Okay, we need to move on. We're a little bit slower than I expected. Right. Do you all understand how competitive inhibition works? Okay, the important bits are you should know how the parameters are changed or not changed, and you should be able to explain why that happens. Okay, good. Let's move to non-competitive inhibition. Non-competitive inhibition, as the name suggests, means that the inhibitor does not compete with the substrate. The binding of the two is completely independent. Okay? So whether a substrate is bound or whether the inhibitor is bound, it does not affect the binding of the other one, unlike in competitive inhibition. Okay? So the binding itself are completely unrelated. The probabilities are unrelated. Okay? That's what non-competitive inhibition is. Most often, well, probably almost always, I guess, well, I guess it has to be like that, they bind to different places on the enzyme. Okay? And we have a name for this kind of inhibition. Allosteric. Okay? Allosteric means binding to a different place. Okay? So non-competitive inhibition is usually allosteric. Okay? Now, when the inhibitor binds, the non-competitive inhibitor binds to the enzyme, it usually changes the conformation of the enzyme, which means that the enzyme usually can bind the substrate, but the reaction doesn't happen. Okay, so it's not really about binding the substrate. It's about the stopping of the catalysis. Yeah.
Is non-competitive inhibition always allosteric?
I guess it has to be, right? Because otherwise they would compete for the same binding site. I think it has to be allosteric. Yeah.
Doesn't the allosteric change the shape of the enzyme?
That's why I said the conformation is changed.
Yeah.
Yeah. The shape of the enzyme is changed. Yeah.
Can competitive inhibition be allosteric as well?
It can. It can. So imagine that the question is whether the question was whether competitive inhibition can also be allosteric. So is it possible that there is a competitive inhibitor that binds to a different site, but at the same time competes with the substrate? Well, it can. So imagine that this is our active site where substrate binds normally, and we have our inhibitor, which looks something like this. Okay, so it binds to a different site, but it will still compete with the substrate for binding. Okay, so it is possible, but it's not. Well, actually, there are some drugs that act like that, but yeah, it's possible. Okay, but it's not an important part of the analysis, but it's possible. Right, non-competitive inhibition. What are the parameters going to look like? What is Vmax going to look like? Okay, well, I still have two people answering, so let's get the middle. Okay, we have one at the front and one at the back. So let's get the middle. Say something. What do you think? Imagine we have an inhibitor that binds independently of the binding of the substrate. The two just do not. What will happen to Vmax? What will happen to the speed at infinite, the rate of reaction at infinite concentration? Okay, so the suggestion will stay the same. Do we have other suggestions?
Go down.
It will go down. Does anybody think it will go up? Nobody thinks that. Okay, so there are two possibilities that it will go down, it will stay the same. Okay, so those who say that it will go down, can somebody explain why it will go down? Yeah?
I think it will go down because now you have you can have an infinite amount of substrate, but at the same time you can have an infinite amount of inhibitors because they bind in different sites.
No. We don't have an infinite amount of of inhibitor. Okay, the amount of inhibitor is finite and stays the same during the analysis. Okay, this is very important. Okay, so we have a fixed amount of an inhibitor and we increase the concentration of substrate. So yeah, do you wanna?
Yeah, I think the Vmax decreases because the non-competitive inhibitor just binds to the enzyme, but it doesn't need to be a specific site. So while the substrate tries to bind to the active site, the inhibitor just binds somewhere, and therefore there is no chance for the substrate to bind.
Not really. I mean, first of all, the non-competitive inhibitors usually bind to a specific site. They do have a specific site. It's only a different site than the substrate binds to. And the two do not interfere. Again, in non-competitive inhibition, the two bindings are unrelated. Okay.
Because when the conformation changed, the enzyme works slower.
Well, it doesn't work at all. That's why it's an inhibitor.
Even if you have lots of substrate, it doesn't work because enzyme is not functioning well, and so it's not functioning at 100%. So it goes down.
Okay. Vmax is going to go down because we have some molecules of the enzyme always that are going to have some inhibitor bound, okay? It doesn't have to be a huge proportion of those enzymes, but there will be some inhibitor bound. Now, if we increase the amount of substrate, it doesn't matter because the two do not compete for each other, for the binding of each other, okay? They do not compete. So even if we have infinite amount of substrate, there will always be some molecules of the enzyme that will have the inhibitor bound. So unlike before, when basically the more substrate we had, the less inhibition we had because they were competing. Now they do not compete. So no matter how much substrate we add, some of the molecules will always be inhibited. And that's why Vmax is going to go down, because effectively we're removing some of the molecules of enzyme from the reaction. Some of them just do not do anything. And that's why, because we said that Vmax depends on the amount of enzyme, it means that Vmax has to go down. Okay, so Vmax goes down. What about Km? Yeah.
No, it stays the same.
Km stays the same because the affinity is not changed in any way. Again, they do not compete for the same binding site. The two bindings are completely unrelated, and therefore there's no reason to change the affinity of the enzyme, even the apparent affinity. So Km in non-competitive inhibition stays the same, and Vmax goes down. So we draw the curve. I'm not sure it's going to work, but sometimes it does, sometimes it doesn't. Okay? Km describes the probability that the ES complex is going to be formed. Okay? Now the probability that ES complex is going to be formed is not affected by the fact that the inhibitor binds. Because the two binding, the binding of the inhibitor and the binding of the substrate do not affect each other. They happen independently. So the affinity is not changed in any way. Only the complex in which the inhibitor is present is not going to be catalytically active. It's not going to form the products. That's all that happens in the inhibitor complex. But it doesn't influence the binding. Yeah?
Why is it as the same as Km of the competitive, not the Km of the regular ones?
It has the same Km as the native enzyme, as the uninhibited enzyme.
Okay.
Okay, let's see if that worked. So this is going to be our Vmax non-competitive. Okay, and if we take our Vmax non-competitive divided by 2, we almost get to the same Km. Okay? Does it make sense? So Vmax is going to go down. Therefore, Vmax over 2 is going to go down, obviously, because that's how it's defined. Okay? And then if we take the substrate concentration at which our new Vmax half is reached, we get the same Km as for the native enzyme. So that's for the non-competitive inhibition. Okay, we're running late a little bit. So let me just finish with the uncompetitive, and then we'll talk about having a break. Okay? Uncompetitive inhibition is a very unusual. Do you have any questions about non-competitive? Is it clear how that works? Raise your hand if you want me to spend more time on that.
Can you just go over why the Vmax goes down?
Say again?
Can you go over why the Vmax goes down?
Okay, the Vmax goes down because no matter how much substrate we add, some of the molecules of the enzyme are going to be inhibited in the presence of any amount of inhibitor. Therefore, they are removed from the reaction, and we can never reach Vmax because some of the molecules are doing nothing. They're inhibited. Okay, uncompetitive inhibition, or anti-competitive, whatever you want to call it. It is a very unusual type of inhibition. There aren't actually that many molecules that use this kind of inhibition, but I think it's quite interesting because it explains other possibilities. In uncompetitive inhibition, the inhibitor binds not to the native enzyme, to the enzyme without anything bound to it, but it actually binds to the enzyme substrate complex. So a substrate has to be bound to the enzyme in order for the inhibitor to bind. Okay? So let me just add. So this is what happens in uncompetitive inhibition. The inhibitor only binds to the enzyme substrate complex. Right, what happens to the parameters? Let's think about it, okay? What will happen to Vmax? What will happen to the rate of reaction at infinite concentration, yeah?
I think it will decrease because now you have it will stop the reaction if it's inhibited.
Okay, correct. Vmax is going to go down because at infinite concentration of substrate, everything is going to be in the ES complex. All of our enzyme molecules are going to be in the ES state. Okay? Does it make sense? Yeah? There's so much substrate that all the enzyme has bound some substrate. And since we know that the inhibitor binds to this complex, then in this situation, at least some of it is going to be inhibited. Which means that some of the molecules are out of the reaction and Vmax goes down. Does it make sense? Raise your hand if it makes sense. Okay, very good. Now, yeah?
At first you have the constant Vmax, but after the
No.
form you have
Vmax is the rate which is reached at the end of our experiment, okay? After adding infinite amount of substrate. So you can't say at first it's something. No, Vmax is something that we get to after adding infinite substrate. We don't start with Vmax, we end with Vmax. And the Vmax, the maximum rate, is going to be decreased because in this reaction, some of the enzyme substrate complexes are going to have the inhibitor bound to it. Okay? Right. What happens to, yeah?
Does that mean then the affinity stays the same?
Well, that's my question. What happens to Km?
In Km it decreases. Affinity.
Why does it decrease?
It decreases because no, the affinity increases because like the ES just binds to it.
Okay, so there's a suggestion that affinity increases, so Km decreases. But I wasn't sure about the explanation. Are there other people who think that Km is going to decrease, so the affinity is going to increase? Okay, not that many. Okay, I'll explain it because we need to get going. Okay, it's true that Km is going to go down. It's going to decrease, which means that the affinity, the apparent affinity of the enzyme is going to increase. Why is that? Well, remember that I said that we can imagine Km, or the affinity, as being the probability of the formation of the ES complex. Now, we have this reaction of ES complex with the inhibitor forming ESI complex, which is basically constantly sucking away the ES complex that we just formed. So the equilibrium between E plus S and ES is going to shift towards ES. There's going to be more ES being formed because some of it is being removed from the reaction. Okay? And this means that the apparent affinity of the enzyme to the substrate is going to increase and Km is going to decrease. Okay? So the curve is going to look like, I'm not sure which color to use. So the curve is going to go up faster than before, but then it's going to reach a lower Vmax.
And that is basically the Chatelier principle?
Correct, yeah. Okay, so this moving of the equilibrium is Le Chatelier principle, exactly. Okay, so if we keep taking away the product, the equilibrium shifts towards the product. Huh?
Keep taking away the product.
Yeah, if we with another reaction we remove it, the reaction goes more and more towards the product. Okay, so the equilibrium is reached basically later. Okay, and this is what it's about. Right. Do you need a break? Okay, some people do. Let's take about 3 or 4 minutes, okay, and then we get going again. Okay. It sounds like a lot of you have questions about a lot of this. Unfortunately, we don't have time in this lecture to answer all of them. But obviously, if you come and see me, we can talk about the specifics, and I can explain those things again. Okay? So. Can't really answer them now, but later on we can, or after this lecture, whatever. Right. In the second part of in the remaining part of the lecture, I want to talk about specific examples of inhibitors that we can look at, and we can talk about whether they are competitive, non-competitive, what actually happens in the enzyme when they bind. Okay. Now, the class of inhibitors that I want to talk about are related to a molecule that we find in our body. It's a neurotransmitter. It's a molecule that transmits signals in the nervous system, and it's called acetylcholine. Okay, this is what acetylcholine looks like. Okay, at this point, you don't have to remember it, but soon you will. Okay, so this is acetylcholine. This is the acetyl part, and this is choline. Acetylcholine is used both in the central nervous system and in the peripheral nervous system to transmit signals. In the central nervous system in the brain, among other things, it's important for the recollection of memory. Okay, so some of the memory circuits depend on acetylcholine. Okay, in the peripheral nervous system, one of the main functions of acetylcholine is transmitting signals from the nerves to the muscle. The neuromuscular junction is dependent on acetylcholine. So all our movements depend on proper functioning of proper production of acetylcholine. Okay, I don't want to go into too much detail about neurotransmission because we're going to talk about it in a month or so. Okay. Now, when acetylcholine is released by a neuron, either on the neuromuscular junction or in the brain, it elicits a signal in the next neuron. Okay, that's the principle of synaptic transmission. So it causes a signal in the next, in the following neuron, because it binds to receptors, etc., etc. Now, obviously, after that happens, we need to get rid of this acetylcholine so that the next neuron can be ready for the next signal that comes. Okay? So the firing of one neuron produces acetylcholine. We have to remove it so that we can have another signal come. Does it make sense? Right. There are many ways how a neurotransmitter can be removed from the synaptic cleft. Yeah?
Can you please repeat that?
Okay. That's a general principle of neurotransmission. Okay? We release a neurotransmitter. It causes an effect. And then we have to remove it so that it can be released again and a new effect is caused. Otherwise, it would just keep causing the effect, which we don't want. We want discrete signals, okay? Right. Many ways of removing a neurotransmitter. In the case of acetylcholine, what happens is that it's broken down. It's degraded in the synaptic cleft, okay? It's broken down here into acetic acid, okay? And choline. And the choline is taken up and the acetic acid as well. The enzyme that catalyzes this reaction is called acetylcholine esterase. Acetylcholine esterase. Okay, it's an esterase because it hydrolyzes an ester bond. Okay? Now, I want to talk about inhibitors of acetylcholine esterase because they're used in medicine and other areas as well, as we'll see in a minute. And they are quite a good example of inhibition in pharmacology, enzyme inhibition of pharmacology, in the pharmacology. Acetylcholine esterase inhibitors are used, first of all, to treat a disease that manifests by impaired memory. Does anyone know what disease I'm talking about? Say degenerative Alzheimer's disease. Okay, Alzheimer's disease. The major symptom is losing memory or being unable to recall memory. A large part of the problem is degradation of neurons that use acetylcholine in the brain. So we can use drugs that enhance the production of acetylcholine, or rather, okay, I've skipped a step, I guess. What happens if we inhibit acetylcholine esterase in the synapse? Well,
will build up.
Yeah, that could happen, but what I'm after is it's going to increase the amount of acetylcholine in the synapse, okay? Which will cause continuous firing, okay? Does it make sense? If we inhibit acetylcholine esterase, the acetylcholine that's there will stay there longer, so the signal that is produced is larger. Make sense? Very important thing because we're going to build on that, okay? So, in Alzheimer's, if we have too little neurons producing acetylcholine because they're dead, okay, we can sort of remove the symptoms by adding an acetylcholine esterase inhibitor that with fewer neurons firing produces the same result. Okay, I'm massively simplifying, but that's the principle. Does it make sense? Why we use acetylcholine esterase inhibitors in Alzheimer's? Yeah? Okay. In the peripheral nervous system, there is a disease called myasthenia gravis. Anyway, you don't have to remember it now, but which is basically muscle weakness. Well, that's what it stands for, a severe muscle weakness, which is caused by the destruction of acetylcholine receptors on the muscles. Okay, so the receptors that normally receive the signal are destroyed by the immune system, and therefore the muscles don't work properly. Okay, and those patients who come, usually they get fatigued very, very early. A typical symptom is that their eyelids, they're unable to keep their eyelids up, especially later in the day, like in the afternoon. In the morning, they're usually fine, but in the afternoon, they're just unable to do that because there's not enough acetylcholine to properly do that. Okay, without going into details. Again, adding an acetylcholine esterase inhibitor potentiates, amplifies the signal that we already have there, even though there are fewer receptors. By having more acetylcholine in the synapse or in the neuromuscular junction, we get the same signal. Does it make sense? This is just a tiny little bit of neurology just to understand why I talk about these inhibitors. Okay? Now, let's look at the biochemistry of it. The active site of acetylcholine esterase contains a key amino acid, which actually catalyzes the reaction, which is serine. Okay, so in the whole protein, this serine residue is the thing that is in the active site and is the most important for the catalysis. How does it work? Well, this, huh?
Where is it?
In the enzyme, in the active site. Acetylcholine esterase, that's the enzyme we talk about, okay? In the catalysis, what happens is, it's a little bit more complicated than that, but anyway, this oxygen attacks this carbon, okay? And choline is released. Okay, so this bond is broken. Choline is released from the enzyme. And the acetyl group stays on the serine. Okay? So in the first step of the catalysis, what happens is that we get a modified enzyme in which the active site serine has the acetyl group bound to it. Okay, and choline, the rest of the molecule, is released from the enzyme. Okay? In the second step, a molecule of water comes in and hydrolyzes this bond, producing acetic acid and regenerating the enzyme. Okay, now you look at me like I had no idea what I just said. Raise your hand if that makes sense. Okay? Okay, about half the people, but okay, I'll have to take that. So this thing, okay, water molecule comes in, it breaks this bond. This thing, which is basically acetic acid with a bit of this water, is released as acetic acid. Huh? And the serine that we started with is regenerated. So we can start again with the same process. Here. Huh? Well, yeah, in the first step, we take this acetic group from acetylcholine and put it on the enzyme, and in the next step, we release this and regenerate the enzyme. Okay? It's actually a very common mechanism, a very common catalytic cycle, that it happens in 2 steps. Some reaction happens first, the enzyme is changed, is modified by the reaction, and then in the next step, the enzyme is regenerated and can return and can catalyze another reaction. It's a very common mechanism. Yeah.
Is this regular hydration?
Huh?
Is this regular hydration?
Well, it's not really hydration.
Hydrolysis?
It's hydrolysis.
Yeah.
Okay. It's not hydration.
Yeah.
Quite a big difference. Okay. So yeah, it's a regular hydrolysis. Okay. There are some other amino acids that help it happen, but I'm simplifying.
So that is not serine anymore, it's something else.
It's acetylserine. Correct. Okay? And then we release it from the enzyme and we regenerate serine. So this is the catalytic cycle of acetylcholine esterase. Now,
Does the water come in the first or the second reaction?
it comes in the second reaction, in the second step. Okay? Right. That's not really the main point, but anyway, it's useful to describe what happens next. Right. As doctors, we want to, in those situations that I mentioned, we want to inhibit this enzyme in order to increase the amount of acetylcholine. Now, one way of inhibiting the enzyme is by basically stuffing the active site with something else. Okay? So producing a molecule that looks sufficiently like acetylcholine, but doesn't actually undergo this reaction. Okay? A simple competitive inhibitor. Okay? Comes in the active site, stays there for a bit, and then is released, and therefore competes with acetylcholine. Yeah? Does it make sense? Okay. I'll show you, I'm going to draw a structure of one of those inhibitors, just in order to tell you what they actually look like. Competitive inhibitors, and I sort of implicitly now mentioned it, very often look very much like the substrate. Either they look like the substrate, or they look like the transitional state, the activated state of enzyme. Does it ring a bell, transitional state? Not really. Okay, in enzyme catalysis, some of you mentioned activation energy. The activation energy is the energy that is needed for the 2 substrates, for the 2 reactants, or the substrate and the enzyme, to get to the activated state. And then once they are in the transitional activated state, they can then drop down to the result. Okay, this was probably in the lecture on kinetics, so I can't really spend time with that. Anyway, competitive inhibitors usually look like the substrate in some way. I'm going to show you one of them, which looks like this. Okay. This drug is called edrophonium. It's called edrophonium. And it's used for the diagnosis of myasthenia gravis. Okay? Why is it used for diagnosis? Imagine a patient comes to you and says, well, I'm really weak and I can't really keep my eyelids or my eyes open or whatever, and I get, progressively fatigued more during the day. There are many possible reasons for that. And if you want to find out whether it's really caused by the destruction of acetylcholine receptors, you can give the patient a very short-acting acetylcholine esterase inhibitor, by which you increase the signal for a short period of time, increase the signal in his body, or his or her body, which means that the symptoms go away. But it only happens if the reason is myasthenia gravis. So it's used for diagnostic purposes. Give the patient a shot of this, and suddenly he improves, which means that he or she has to have myasthenia gravis. If it does not improve the condition, it's probably something else. Okay? It's used for diagnosis, very short acting.
Side effects?
Huh? Sure, there are side effects, but I don't really have time for that. Okay? So very, very short acting, just a few minutes. Now, can you see the similarity with this molecule? No. Okay. Let me show you. Well, actually, we can go all the way here. This is the same bit. Okay, there's some additional stuff here, there's the ring there, but this backbone of the molecule looks very much like acetylcholine. Well, like choline anyway. Okay, can you see the similarity? Okay, so this is a typical, very short-acting, perfectly reversible inhibitor of acetylcholine esterase. It goes in the active site because it looks similar to choline. It sits there for a bit, but obviously no reaction is going on because there's nothing to remove or do, okay? So, typical competitive inhibitor. Now, we can, and obviously we could make lots of molecules like this, and there are lots of them. For example, there's a drug called tacrin, which is used for Alzheimer's. It's also a competitive inhibitor of acetylcholine esterase. It kind of looks like acetylcholine, but much less than edrophonium, so I'm not going to draw it because the similarity is not that great. Okay, so it's not for teaching purposes, not that useful. But tacrin is, well, now it's a bit less used, but it used to be one of the major drugs for Alzheimer's. Okay, it's much less used now, but anyway. Okay, so tacrin, probably a good idea to remember that as well. Right. However, based on the catalytic cycle, the mechanism of acetylcholine, we can use a different strategy to inhibit the enzyme. We can produce a molecule that looks like acetylcholine, but instead of the acetyl group, which is very easy to remove in the enzyme is built to remove this acetyl group from serine very quickly, we can use a different group that also binds to serine, but is very, very slow to hydrolyze. Okay? So we produce a synthetic molecule that looks like this, binds to the enzyme thinks, oh, this is acetylcholine, that's fine, let's just do the catalysis. It binds this other group to serine, but then it's stuck and it actually can't do anything else. And if this serine is blocked by some other chemical group, we talked about covalent, non-covalent, okay? This binds non-covalently, just goes to the active site and goes out. Okay? Now we're talking about a covalent modification of the serine. Okay? So once we have something else sitting here, which cannot be easily removed, the enzyme is inhibited. It can't do anything. Okay? Now, since this inhibition by this other group, and I'm going to draw it in a minute, since this inhibition by this other group takes much longer, we're now moving from the perfectly reversible inhibitors more and more towards irreversible. Okay? The longer this new group is stuck there, the longer the duration of the inhibition, and therefore we're moving towards irreversibility. Yeah?
Don't we need, like, at one point, if all the enzymes are inhibited, isn't that something just on the other side, like, isn't that also bad?
Oh, it is, absolutely. And I'll show you in a minute how bad it can be. Okay, let me draw a structure. Again, you're not required to know the structures, okay, of another drug, but I do want you to know the names, okay? This drug is called pyridostigmine. Pyridostigmine comes from pyridine. This is a pyridine ring. Okay, pyridostigmine. And it's also an inhibitor of acetylcholinesterase. Now, do we see the similarity with acetylcholine there? This is actually way more similar than edrophonium. Well, actually, nitrogen shouldn't be there. Okay, there would be an oxygen here. Oh, sorry, there would be a methyl group there. Do you see acetylcholine almost? Okay, but in this case, instead of acetyl group, so in acetylcholine, this would actually look like this. Okay, there would be methyl group instead of this thing. Yeah? But otherwise it looks quite similar. Now, this new group is derived not from acetic acid, it's derived from another acid which is called carbamic acid. So this group is not acetyl, but it's carbamoyl. It's a carbamoyl group. That's, you just, okay, that's how it is, okay? I'm not gonna, it's called carbamoyl. It's a carbamoyl group. So, pyridostigmine comes to acetylcholinesterase. Acetylcholinesterase binds it because it looks kind of like acetylcholine. It tries to break this bond, and it succeeds. It breaks the bond. It binds this carbamic group, carbamoyl group, well, dimethyl, but whatever, carbamoyl group. It binds to its serine, but this carbamoyl group is much harder to remove than acetyl group. So as long as this carbamoyl group is sitting on the serine there, the enzyme is inhibited. Okay? So this is a different kind of inhibition. Now, this is fine, but in the end, the carbamoyl group actually does get removed. Okay? So even though the duration of inhibition is quite long, in the end, the enzyme is regenerated and it can do its stuff again. Okay? So chemists, and they weren't really pharmacologists, were thinking about producing an inhibitor that would be completely irreversible. Okay? They would just block the enzyme forever. Okay? So they were thinking, okay, let's make another group, which is not going to be carbamic acid, or based on carbamic acid, it's going to be based on something else. And they produced this molecule.
How common are the carbamic groups? How
Oh,
do we see these in our life?
Well, yes. When we talk about the synthesis of nucleosides, pyrimidines, and also the synthesis of urea, you're going to talk about carbamoyl phosphate, which is an important intermediate, which is based on this. Okay, so you'll hear about it soon enough. But anyway, it's completely unrelated. Okay, actually, pyridostigmine is based on physostigmine, which was isolated from a plant, so it was actually found in nature, and okay, it wasn't designed. But anyway, the chemists produced this nice molecule. Okay, which looks a bit less than acetylcholine than before, but you can sort of see a similar theme. Okay, this could sort of be our choline, and this could sort of be our acetyl group. Can you see it? Okay, it actually, for the enzyme, it actually looks pretty much like acetylcholine, even though on the blackboard it may not. Okay. Now, this interesting group gets released the same way as before. Gets released from the enzyme. From the molecule. It binds to the serine, and when it's bound there, it basically never leaves. Okay, it's almost completely irreversible inhibition. Okay, so it blocks the enzyme, kills the enzyme, and that's it. Now, this nice molecule is called serine.
Isn't this the gas?
Yeah, it's a nerve gas that is used for killing people. It's extremely effective because it's a gas, so you breathe it in, and it basically immediately blocks your acetylcholinesterase in all your body. Now, how it kills you is something that's maybe a little bit difficult to explain now, but it kills you basically by the fact that if you have too much signals going in your muscles, okay? If you have no signals, nothing happens, your muscles don't move. If you have just enough signals, your muscles move, okay? If you have too much signal, they stop moving again. Not because they're clenched, which is something you might think that will happen. Actually, they get relaxed. There's complete paralysis if there's too much signal. I can't really go into the mechanism now, but we'll talk about it in a month or so, okay, how that actually happens. So what you get, if all your acetylcholinesterase is completely blocked, you get a paralysis of your muscles. And obviously, for your life, there's one important muscle that you need to be, that you need in order to be working. Which muscle I'm thinking about? It's not the heart, actually. Because a muscle. It's a muscle. Exactly, diaphragm. Okay. Because it's only skeletal muscle that is dependent on acetylcholine. The heart doesn't really depend on acetylcholine. Okay, there will be some effects, but for its function, the heart doesn't really need it. Okay, it's only skeletal muscle. And diaphragm is a muscle that is quite important. It's a skeletal muscle, but you need it for breathing. Okay, so you die because you suffocate, and usually you're fully conscious when that happens. Right, so this is serine, and it's also a acetylcholine esterase inhibitor. This group of inhibitors, so if these are carbamates, because they're based on carbamic acid, these are called organophosphates. It's completely synthetic, yeah.
And they started using it as weapons after that?
Absolutely. These were synthesized as weapons. They are also, and actually much more so, hopefully, they are used as pesticides. They're used to kill pests. Pesticide? Raise your hand if you know what a pesticide is. Yeah? Okay. In agriculture, when you want to kill insects or whatever, use a pesticide. These are extremely effective pesticides. Well, they kill people as well, but mostly they're used in agriculture as pesticides. Extremely effective and pretty toxic to the people as well. Okay, so you have to be careful when working with organophosphates because they tend to be deadly. Okay, that's all from me. Do you have any quick questions? Let me just say, because we only had 2 lectures together, when there's a chance to ask a question when everybody's in the room, it's a good idea to ask it because there's a very, very high probability that almost everyone in the room has exactly the same question. Okay? So, but they're too timid to ask. So, if you have a question, ask now because there are other people who will be happy to hear the answer. Yeah?
Did you put out something to read about this? Like
Any literature?
this is pretty much.
Well, the question is about literature. For the second part of the lecture, the only literature are pharmacology textbooks, which is not something I really want you. Well, I don't want to make you read pharmacology textbooks because you'll still have time for that. Okay. For the first part of the lecture, there are a lot of very good materials, and I can probably link to some of them, okay, about the analysis of inhibition. It's usually related to enzyme kinetics, so basically a continuation of enzyme kinetics, you get the inhibitors there as well. Okay, so I'll try to link something. Maybe it's already there, I don't know.
Yeah, but where can I catch the, because the second part I fell out.
Okay.
We watched the video.
Yeah, you can watch the video, obviously, which hopefully I'm going to put up very soon. For the pharmacology part, what I want you to remember, obviously, I would like you to be able to explain the mechanisms behind it, okay? But really, what you're going to need for the test, which is something you're interested in, presumably, are the names of the drugs that I mentioned here, okay? And what they do, okay, that there are acetylcholine esterase inhibitors and whether they are reversible or irreversible. It's basically all I would much prefer that you remember the principle, but you will need to know the names as well. Yeah.
For the normal acetylcholine esterase, it's the acetyl bond that binds to the serine.
Correct.
But for the serine, it's actually not the organophosphates.
It is. It's this whole thing. Yeah. This is what binds to serine. Okay, it binds in a slightly different way. It's not quite an ester. It's a bit slight. The fluorine goes away. It's slightly more complicated, but this is what binds to it. Yeah. Okay, if there are no more questions, that's it for today.